Hello, everyone, and welcome to No Awful GCSE. If you are here, really well done, and if you're going to join soon, also well done. So, let's begin. On our schedule today, it says that we're going to be doing biology first between 5 a.m. to 6 a.m. So, now we're going to be looking at cell biology. So, what I would like you to do is spend 10 minutes writing as much as you can about cell biology and use these prompts, these 12 prompts here, to help you. You can do this on an A4 page, you can do this online, however you find best. I'm going to give you 10 minutes to write as much as you can, and then we're going to go through it together. You may begin now. Just a note here, you now have under 5 minutes to complete this. Okay, so that is now 10 minutes. I also did the same thing, this same blurting activity online. So let's go back to the screen now. I'm going to show you what I did. So I've covered some of the topics. I didn't get to cover all of the topics, but what I want you to do now is get a different coloured pen and we're going to go over what you missed out, because this is what you don't remember, and this is quite important. So first things first, eukaryotes and prokaryotes. What you needed to get there was how animal and plants are eukaryotic, and they have a cell membrane, a cytoplasm, and a nucleus. And bacterial cells are prokaryotic, and prokaryotic cells have a DNA that's not included in a nucleus, but in a plasmid and loop. Then we need to talk about animal and plant cells and what they each contain. Again, animal and plant cells have cell membrane, nucleus, ribosomes as the general structures, and then chloroplast, cell wall, and permanent vacuole as the specific structures. You could have described them as well. So that's what I missed here. The nucleus contains the DNA for coding, and the nucleus is also used for controlling the functions of the cell. Ribosomes are where protein synthesis occurs. Now, chloroplasts are where photosynthesis takes place because of the green pigment called chlorophyll. The cell wall contains cellulose, and it strengthens the cell so that more water can be uptaken by osmosis. And the permanent vacuole contains cell sap, which helps increase the rigidity of the cells. Then we looked quickly at specialized cells, which are cells that undergo the process of differentiation of gaining new subcellular structures in order to be suited for its role. So we looked at sperm cells. The function of sperm cells is to carry DNA from the male, carry the male's DNA and fuse it with the female's DNA. So we talked about how it has mitochondria crammed in its head. We discussed how it's streamlined, it has a long tail, and also what's necessary is the achromosome. So the achromosome is what you call the top of the sperm's head, and this contains a lot of enzymes which break down the outer layer of the egg. Then we have the nerve cells. The nerve cells transmit impulses, electrical impulses, and there's a long axon, and the axon is what we call this body here. This is a long axon, which means that the impulses can be carried long distances. It has a lot of extensions from the cell body. The nerve endings have many mitochondria. So again, lots of energy in the mitochondria. So if you didn't get any of those, make sure you write that in a different color so you remember. And finally, we looked at muscle cells, which are specialized to contract and relax really quickly to move bones. They're called striated because they're in this strained pattern, and they have special proteins which help slide over each other. There's lots of mitochondria for energy. And also they can store a chemical called glycogen. And glycogen, which we'll look at later, can be easily converted to glucose, which can obviously be converted to energy. So that's really useful. Okay, so you can already see how much we've missed off from here. Now we're going to look at the plant cell special cells. So we can begin with xylem cells. Xylem cells are specialized to transport water up the transpiration stream. Now, upon formation, there's a chemical called lignin, which eventually leads to the cell's death. And because they're spiraled up in the cell walls, it means that they have very strong cell walls, and this can withstand the water pressure that's constantly going up. That's all we need to know about that. Now, phloem is specialized to carry the products of photosynthesis from the leaves where they're made to around the plant. they have these things called sieve plates, which help to direct the flow of different substances because it's a multi-directional flow. And despite losing many subcellular structures, the energy needed for the cells, for these phloem cells to be alive, comes from neighboring cells. So it gets a lot of energy from neighboring cells. And finally, root hair cells. So root hair cells are specialized to take up water by osmosis and minerals by active transport. It has a large permanent vacuole, which is used for osmosis, and it's useful for osmosis. It has a large surface area due to its root hair, meaning more water can come in, and also there's lots of mitochondria. Okay. So then our next one was looking at microscopes. So I said that there's light microscopes and electron microscopes. Light microscopes have lower magnification and lower resolution, and electron microscopes have a higher resolving power. That's practically all you need to remember. Then we looked at mitosis. So probably need to explain this a little bit better than that. So the first stage of mitosis is the longest stage, and this is when all of the subcellular structures in the cell duplicate. So all the chromosomes duplicate, subcellular structures duplicate. This is the longest stage because it takes very long. So the chromosomes become X-shaped. That's when that happens. Sorry, I don't know why it keeps going. Then the second stage is when the chromosomes and other subcellular structures are pulled to either side of the cell. Almost, you can see the beginning to form two different cells here. And this is relatively quick. And then the final stage where the cell membrane and cytoplasm split, forming two identical daughter cells. So mitosis is a process in which one cell becomes two identical daughter cells. So it's a form of cloning, you could say. So after, let's see what else I missed. So I missed stem cells, diffusion, osmosis, and active transport purely because of time, but we can quickly go over those as well. So let's go over stem cells. So So stem cells are undifferentiated cells. What this means is cells that haven't differentiated, haven't specialized yet. And they're undifferentiated cells, which can either, one, give rise to more stem cells, so they can divide to form more stem cells, or two, they can produce any type of cell. So what happens is we have the different stem cells. We have the embryonic stem cells, which are found in the embryo. The ball of cells are actually the ball of stem cells. And these then differentiate to form all the cells we need to survive. We then have adult stem cells. Now, adult stem cells are found in the bone marrow. The reason why it's found in the bone marrow is because many of the blood cells are unable to divide. Just how we talked about mitosis, many of the cells aren't able to undergo mitosis or meiosis. Therefore, they need stem cells to create these blood cells. all the blood cells refer to platelets. So these are what the adult stem cells make. What happens is the stem cell breaks every time it divides by mitosis. It forms another stem cell and then a blood cell. That's how it maintains its population, as well as giving rise to cells that are unable to form. Okay, and we've talked about stem cells, and now let's just talk about transport. So diffusion, osmosis, and active transport. Now there's also a required practical hidden here, so within the osmosis part. So diffusion. Net movement of particles from an area of high concentration to an area of low concentration, moving down the concentration gradient. Now, diffusion can be for anything, and an example of diffusion is in the red blood cells gaining oxygen. Okay, that's diffusion. Now going on to osmosis. Osmosis is the net movement of water through a partially permeable membrane. So all these substances can hit it, but only water can go through because it's a partially permeable, so only water is a small enough molecule. And finally, active transport. Active transport is a net movement of particles from an area of low concentration to an area of high concentration. Because you're going against the concentration gradient. So overall, we can see that diffusion is a passive process. It does not require energy. Osmosis is a passive. And active transport, however, is active, as the name suggests, which means energy is required for it to work. Now we have a required practical in osmosis, so there's a bit more information that we need to know about osmosis. So osmosis has different forms. There's hypertonic, hypotonic, and isotonic. You don't necessarily need to know the last one, but the first two you definitely need to know. So these are what you describe different solutions. So hypertonic is when the inside of the cell has a higher amount and the surroundings have a lower amount. Therefore, water moves from an area of high concentration to an area of low concentration outside of the cell. That's what hypertonic is. Now, hypotonic is when the interior of the cell has a low concentration of water and the outside has a high concentration of water, and therefore water moves from outside in. Now you can imagine isotonic, therefore, is when there's an equal amount of water both on the outside and the inside, so there's no movement. So they're just some key vocabulary you need to know. And then finally, we're going to go through the required practical. For osmosis. So you begin with 3 different solutions. Excuse the drawings. And each solution is going to have a different volume, a different concentration. A different concentration, it can be of sugar and water, or it can be of salt and water, whatever. So one is going to be pure distilled water, which means no sugar in it. And then the other 2 can be, for example, 0.5 molar solution and 0.1 molar solution. So what this means is this one has the least sugar. Well, this obviously has the least sugar, the pure distilled water, but then it's a 0.1 mole and then it's a 0.5 mole. So they all have different concentrations of sugar and water. And then what you're going to do is you're going to get a potato And with that potato, you're going to use a core borer, so which is just this metal thing. And you can use a core borer to get 3 cylinders of potato. Now, the independent variable, what you're changing here, is the solutions, the different concentrations. The dependent, what you're measuring, is the change in volume of the potatoes. And the control variable, what you're keeping the same, you need to keep the length and mass of the potato roughly the same. So what you need to do is now you have your 3 cylinders of the potato. They may all be different sizes, so you need to cut them to the length at which the smallest potato is, so that they're all the same length. And also make sure that the reason we're using the core borer is that they all have the same diameter. Now once you've done this, you need to put these 3 into these 3 solutions and leave overnight. Oh, and also measure the mass before you put them in, because we're measuring the change in mass. Then in the morning, you're going to measure the new mass. So you take it out of the solution, you gently use a paper towel to dry it, and then you measure the mass. Now there should be a change in mass, but using the knowledge we know, the greatest change in mass, your distilled water, because there's the greatest concentration difference. So there's the most water outside, so lots of water will come in, so it'll be a greatest increase. Then the 0.5 molar will have the least increase because it was mostly sugar, so it was similar. This concentration is similar to the inside of a potato anyway. So it was isotonic conditions. So that is cell biology revised. That's the amazing thing about blurting. You already know the information, you just have to output it onto a page. Of course, this is the summed up version, but within the exam, a lot of the points are just the key basic points that aren't going into too much depth, really. One more thing that we did not mention that I can put here in green is about therapeutic cloning. So therapeutic cloning involves an embryo being produced with the same genes as the patient. So what happens is an embryo is produced, and this then could be harvested to take the embryonic stem cells from these embryos, because they are very useful in science. And the advantage is that they would not be rejected by the patient as they have the exact same genes. Okay. Let's review that now. So hopefully, if there was anything you did not get, then you definitely wrote it down and took notes. But now we're going to move on to the next topic, which is... Oh, sorry, I forgot to show you this. Organization. So I want you to do the same thing. I'm going to set you a 10-minute timer. So by 5:40, I want you to spend 10 minutes writing as much as you can about organization and use the 13 prompts to help you there. I'll leave you to do this now. Okay, so thank you for doing this activity. We're now going to look at the board that I did about organization. I didn't get to completely finish this topic. However, we can go right for the topic through what you've missed. So let's start. The first topic was principles of organization. Now, what this just means is how a cell is made up of a tissue, is made up of a group of specialized cells, and tissue then creates organ. Muscle is not really necessary, and then organs create organ systems, such as the digestive system, and then organ systems create organisms. Then we looked at the digestive system. So I made a little diagram here, and it's made up of the salivary glands. So first, food goes down the salivary glands. The number system is in relation to the enzymes, so which breaks down starch. So this enzyme is prevalent here, which breaks down starch. Then you have the food go to the stomach. The stomach contains hydrochloric acid. The hydrochloric acid kills any bacteria and provides the optimum pH for protease enzymes to work. And then food goes to the small intestine and then to the large intestine. And in the small intestine is where all the enzymes start to work. So you also see carbohydrates and lipids. Lipids come in from the pancreas. Lipase, the enzyme lipase is made in the pancreas. And then we have bile. Bile is not an enzyme. Bile is just a liquid that emulsifies fats, which just means converts it from large droplets to small individual droplets. And it's made in the liver, stored in the gallbladder, and also maintains optimum. Now, that brings us on to more about enzymes. So enzymes are biological catalysts that increase the rate of the reaction without being used up. And the lock and key theory states that an enzyme will only bind to a substrate, which is whatever it's breaking down, as long as it's complementary to the shape of its active site. So if the substrate had a different shape, then it would not be broken down by the enzyme. Therefore, we can call enzymes specific. Now, enzymes also have certain conditions that they can work in, and this is what I described as temperature and pH. Now, they both have graphs that look like this for temperature and pH, which just means at here and here are the optimum conditions where the enzyme works at its best, and then beyond the optimum conditions, the enzyme then denatures, which means its active site changes, and therefore it can no longer work. So that's lock and key hypothesis. Then the next topic we looked at, let's just check back into that. So the next topic was food tests. So I could not really remember regarding the food tests. GCSEs were a long time ago for me. So here are the different ones. I'm going to use my red pen because I forgot. So we have lipids, starch, proteins, and sugar. I think those are the 4 that we're testing for. Yes, that's correct. So yeah, Benedict is used for sugars and it turns brick red to green, a variation of different colours. Burette solution is used for proteins and it turns it purple. Iodine is used for starch. And if it turns from orange to blue-black, then there is starch present. However, if it remains orange, there's no starch. And for lipids, we use... Oh no, sorry, there is no test for lipids because it's the emulsion test, emulsion test. So this is when there should be a reaction between the lipids and ethanol, and it produces a white emulsification, so like almost cloudy white on top, and then you know that there's lipids present in there. So because I forgot that, I'm going to just relay it back to you. So we have lipids, which are tested by the emulsification test. So emulsification and ethanol. We have sugars, which are tested by the chemical of Benedict's solution. and turns to red. We have protein, which is tested via burette and turns purple. And we have starch, which is tested via iodine, and if present, turns from orange to black or blue. So they are the food tests. Now, let's revise the final bit. So we looked at the heart and the blood vessels. So I didn't get too far into this, but this is the heart diagram. I'm just going to double check that this is correct. I don't want to tell you wrong information. But there is a way, because it's quite easy to forget the different parts of the heart, and therefore there is a way that I learned this. Okay. Okay, we got it right. So, a way I learned this, if you struggle to remember the parts of the heart, is first of all draw the diagram like this. So there's always something coming in, there's always something coming into the heart, and there's always something leaving the heart. And then just write V A V A V A V A. And then you need to know that pulmonary is anything to do with the lungs. So pulmonary vein, this is called the pulmonary vein because it's bringing the oxygenated blood back. And this is pulmonary artery because it's bringing blood to the lungs. So you know that these two are the pulmonaries. And then you know that it's always opposite on a heart. So this is the right atrium and right ventricle, and this is the left atrium and left ventricle, even though it's the opposites to what we're looking at. And then we just have this one and this one left, and you'll learn that the only two left will then be vena cava and the aorta. They're the two odd ones out. And that's how I learn the structures of the heart. So the deoxygenated blood comes in here, and the oxygenated blood comes through here. The oxygenated blood is going to different organs and going to give the oxygen, and the deoxygenated is going to get oxygenated. Now, it's important we learn the different types of parts of the heart, and I didn't write this down, so let me write it now. So we have capillaries, we have veins, and we have arteries. So veins carry deoxygenated blood. Arteries carry oxygenated blood to the body, so from the heart to the body. And the capillaries are just this thin, very fine network which gets minerals and different dissolved substances to diffuse through into the bloodstream. Okay? Because remember, in the blood, it's not just blood, we also have plasma. So we can go through plasma now. But just one more thing. So arteries, because they're carrying the oxygenated blood, because in the red blood cell, we have this chemical called hemoglobin. And what hemoglobin does is it combines with oxygen to form oxyhemoglobin. So what happens is the oxygenated blood is therefore heavier because it has this new combined chemical. So as we've discussed, the oxygenated blood is heavier, so arteries have more pressure to deal with. And because of this increased pressure, they have elastic fibers in the cell walls. And what this does is it allows the arteries to stretch out just a little bit so that the blood can go. These pulses are determined by the pacemaker, which is found in the atrium or something like that, I believe. Now, as we said, veins, so arteries are away from the heart, veins are towards the heart, so to the heart, therefore to the lungs, and they are very thin to allow the low pressure blood to go through. Now, it's important to quickly discuss what valves are and the purpose of valves. So valves are these things which are found here and here, and they have a lot of different names: semilunar, lunar, atrioventricular. But I think you just need to know the functions of the valves. That's the main bit of importance. So valves, what they do is they prevent the backflow of blood. So imagine a door that constantly swings open, but as soon as the blood passes, it then slams shut. And this means that no more blood can go back, and the backflow of blood can be very dangerous and cause lots of heart problems. Now, talking about the blood and also heart problems, it's important to discuss the blood in this topic. So, as we said, we have something called plasma. Blood is made up of plasma. So plasma is this yellow liquid which carries around all the substances of blood. So can we try and remember what the plasma holds? Yes. So we have the white blood cells, we have red blood cells, and we have platelets. So what are these all individually for? Let's see. So white blood cells are part of the immune system, which we'll look at in the next topic tomorrow. So white blood cells are part of the immune system, and they're part of the body's defense system. They actually have three functions, which we don't have to talk about now, but let's just introduce it. So the first function is phagocytosis, which is engulfing a cell, so it engulfs bacteria. The second is producing antitoxins. And toxins are the things that make us feel ill. So the antitoxins form complementary sites to this and therefore bind and destroy it. And it also forms antibodies. And these are small proteins that clump together against the bacteria. So white blood cells are like that. Red blood cells, on the hand, don't even have a nucleus. They have the chemical of hemoglobin, combines with oxygen to form oxyhemoglobin, and therefore carries that around the blood. You may be asked for some advantages of red blood cells, and you can talk about the biconcave shape of red blood cells. You can also discuss how red blood cells, because of the fact that they don't have a nucleus, that means there's a lot more space for oxygen. And you can also talk about how they have a disc shape, which I guess is the same thing as a biconcave shape. And finally, now we're going to talk about platelets. So platelets are the things that help the blood clot. Platelets are very integral. We need them to happen. So what happens is, let's say a very small artery, or a very small capillary is cut. What happens is this then starts to create a bump and enlarges, and it's almost like the brain is alerting the platelets to come near to that site. And what happens is the platelets arrive, they form a net-like structure working from the outside of the wound in, until it's fully covered and concealed. And there are two advantages to it being concealed. So firstly, it prevents any microorganisms entering, because that would not be good for the wound. And it also prevents blood leaving, because that could lead to blood loss, which could be very bad. And that's the purpose of platelets. Without platelets, we would actually die, because we could just keep. We need to discuss coronary heart disease. So coronary heart disease are just any diseases any diseases to do with the heart. So coronary arteries are things that provide blood to the heart. So these are the really important arteries that provide blood to the heart. So imagine that's the heart. These are really important. So what happens if these really important arteries get blocked? That means then there's no oxygen get heart, and when there's a lack of oxygen, a heart attack can occur. Now, what can cause these coronary arteries to block and build up? Well, this is called the buildup of fatty material, and we call this fatty material cholesterol. Now, everyone has cholesterol, so cholesterol is perfectly healthy, but the volumes of cholesterol, if you have high cholesterol, then it can very much damage you, because it can limit the blood flow to the heart and therefore the oxygen, and cause heart attacks. So we have a couple of solutions which you need to know for your biology spec. So the first solution, you may have discussed this in your blurting, are statins. We also have stents. And then there's also cholesterol blocker drugs. Now, the last one, the cholesterol blocker drugs, are not on your spec, but in many papers across the years, I've seen these come up in questions, so I think it'll be quite beneficial for you to know them. So what are each ones? Statins are these drugs that in the UK are given the moment you turn age 50, and what they do is they decrease the levels of the bad cholesterol, which could lead to coronary heart disease. It's really good. They reduce the chances of having heart strokes, failures, and attacks. Sorry for that writing. And they're really good. However, there's so many side effects, which could be really badly for your other types of health, and also they may not have an immediate effect. Oh, sorry. This is supposed to go there. Now, stents are these metal placements, and what you do is you put these into your arteries. They're inserted into your arteries, and they keep the arteries open to allow blood to flow through. Now, the good thing is it keeps the arteries open, but only in that particular part. Only in that particular part of the artery. Now, if you think about it, if you widen this part of the artery, still all of this part and all of that part is still vulnerable to being blocked by cholesterol, so it's very limiting. However, there is limited side effects, and the cholesterol blocker drugs are similar to stents. However, there's less side effects, and also the moment you take statins, you have to take them for the rest of your life, which is also another thing. Now, what other problems can there be with hearts? There can be many problems with hearts. So we've talked about coronary heart disease. Another thing that's worth mentioning is faulty valves. So a valve is something that, as we discussed, prevents the backflow of blood. So a faulty valve is when your valve stops working. Now, this can just be out of use and old age that your valve stops working, but it could be really dangerous, because if blood flows in the wrong direction, it means that less oxygenated blood is getting to the heart again. So it can be solved with two solutions. The first is a mechanical valve, so getting a metallic valve placed in your heart. However, it has a very short lifespan, and constant medication is needed because your body is constantly trying to reject this metal and plastic thing. It's recognized as foreign. So constant meds required. And then we have a biological valve. Now, a biological valve would normally come from something like pig's tissue. It would last 10 to 12 years and it's quite effective. However, you would also have to take medication, but it will be less medication because it's not as foreign as putting plastic into your body because it's still tissue. So less medication is needed. And finally, the final problem to do with hearts is heart failure. So heart failure, however, can be solved with a heart transplant. A heart transplant requires a donor who has recently died. So these are not always available. That's the issue. Issue number one, they're not always available, and therefore an artificial heart may be used time that you're waiting come. Now let's see what else. Have we missed anything else? Okay, final things, we need to look at cancer and plant tissue before we go on. It's overrunning slightly. Okay, so cancer. There are two types of cancerous tissue. One is when it spreads and forms secondary tissue. Oh, here, it's just come to my head. So we've got benign and malignant. So malignant is the one that spreads and forms secondary tumours. Now, tumours are just a cluster of cells that can't stop dividing and therefore can be cancerous. And benign tumours are tumours that remain within the membrane, remain within the tumour's membrane. Now, cancerous just means spreading. So malignant tumours are cells. Malignant cancer is basically comes from cells that constantly keep dividing. Could it be due to sun radiation or anything really? And it spreads across the body and forms something cancerous, which is very sad and dangerous. And unfortunately, they haven't found something to prevent it just yet. Now, lifestyle risk factors for cancer include smoking, obesity, and UV light, which is light from the sun. And genetic risk factors, you can inherit genes which can increase the likelihood of you getting cancer. And finally, we're going to just talk about plant tissue. So in a leaf, there are the following tissues. So there's the waxy cuticle, the upper epidermis. Let me just write leaf structure. The palisade mesophyll layer. I would use a mnemonic to help remember this. I use a mnemonic Wupsels. The spongy mesophyll layer. The lower epidermis. And the stomata. So each of these have their functions. The stomata contain guard cells, which open and close the pores to allow oxygen and water to escape as a gaseous form. The upper and lower epidermis is clear layers. So the waxy cuticle is a clear layer to allow light to come through. It's important to note that the waxy cuticle isn't found on the underside of a leaf, and the reason why is because the sun can't reach the underside of a leaf, and therefore there's no point. The palisade mesophyll layers are probably the most important. This is where you find chloroplasts, so cells with a lot of chloroplasts, which is what we call palisade cells. You may have learnt this in year 7. And then the spongy mesophyll is basically when there's loads of gaps, allowing for water vapor to reach these cells because photosynthesis requires water as well as light and energy. Okay, now that is all we need to know for organization. So this morning we've looked over quite a lot. So well done if you've been here, and thank you for watching for this part of the series. So now we're going to look at the next topic. So 1 second. Trying to get to the chemistry because we're going to begin chemistry now. Six to seven. So this started a bit later than usual. Sorry for the delay. Okay. Let's begin. So I'm going to give you 10 minutes as usual, and what I want you to do is blurt as much information as you can about atomic structure and the periodic table. Use the red topics below as a guide and to help you with these little prompts. And yeah, just try and get round to doing every single one, and I'm going to do the same on the computer. See you in 10 minutes. Okay, everyone, I'm back, and hopefully you have written as much as you can. I got up until the alpha particle experiment of the history of the atom because I had to do some sorting out of my live stream, but hopefully you got further than that. And let's go through and see whether you got them right. Now make sure you use a different coloured pen when looking at these. The first thing we need to look at is atoms, elements, and compounds. So, atoms have a radius of 0.1 nanometers. I already missed that in my thing, so I'm going to write all my improvements in red. So, atoms have a radius of 0.1 nanometers. The nucleus, as we said, is in the middle of the atom, and it contains protons and neutrons. And the nucleus has a radius of roughly 1/10,000 of the radius of an atom. So the radius of an atom is 0.1 nanometers. Then the radius of the nucleus is 1/10,000 times the radius of an atom. So it's the radius of a nucleus is 10,000 times smaller than the radius of an atom. Now that's crazy. So in reality, the particle model would look like the nucleus like that, and the exterior like that. But anyway, moving on. And then we have electrons, which are found on shells embedded on the outside. Now this is a nuclear model which Ernest Rutherford and Niels Bohr combined came up with. Now, atoms are neutral, as we said, they are neutral. So, atoms can become charged by losing or gaining electrons. Now, elements are when there are loads of different types of atoms, all the different types of atoms with the same number of protons, neutrons, and electrons. So, they're all identical. So, loads of lithium atoms will be called the lithium element, like that. Now, atoms of each element, as we said, can be represented by one or two symbol element, which is presented on the periodic table. Now, it's important to just remember the calculations. So let's look at carbon. Carbon has 12 electrons. The mass number is 12 and the atomic number is 6. Now, when you see this on the periodic table, you may ask, how do I know which number is the mass number and which number is the atomic number? Just think, the mass is always going to be the larger number. So mass is going to be greater than the atomic number. So from that, we always know that 12 is the mass number and 6 is the atomic number. So the mass number is the number of protons. And we know that the number of protons in an element is equal to the number of neutrons, to the number of electrons. Why do we know that? Because we know that the overall charge of an element is neutral. Therefore, the number of balance out and equal the number of electrons. So we know that the mass number is the number of protons, i.e. the number of electrons. Now, the atomic number is just the number of neutrons. Oh no, sorry. I'm getting this confused. Sorry, this is wrong. The atomic number is the number of protons. That makes more sense. And the atomic number is therefore the number of electrons. And the mass number is the number of protons plus neutrons. Therefore, if we do some maths, you'll soon realise that, let's start with another element, for example, lithium 7,3. If you were asked to find the number of protons, neutrons, and electrons in this element, we'll start by saying we know that 3, the atomic number, is the number of protons. We know that the number of protons equals the number of electrons. And we know that neutrons plus protons equals 7. So to get the number of neutrons, you just do 7 minus 3 and you get 4. So that's how you would work it out. Now, an isotope, in chemistry we also have isotopes. So isotopes are different forms of the same element, and these different forms come into being by the fact that they have different numbers of neutrons. And if they have different numbers of neutrons, that means they have different. So let's look at an example. Let's go back to carbon 12. So here we can see that there are two isotopes of carbon. Remember, we said isotopes are different forms of the same element, so the same carbon, but with a different number of neutrons and therefore a different mass number. Now, what you may be asked to do in the exam is work out the relative atomic mass. Now, what that means is overall, most of carbon that might be found may be 75% with 12 as the and 25% with 7 neutrons. So what they want you to do is find out where would the middle be and where would most of the isotopes be around. So therefore you kind of have to find the middle, the intermediary value. So what you do is you times the abundance, which is 75, by the mass number, which is 12, and you add this to the abundance of the other one, so 25 times 13, and you divide the entire thing by the sum of the abundances, which should always equal 100, so 75 plus 25 is 100, and then you should get the mediary value, what should be in the middle. Okay, so that's enough of that. And then finally, just looking at compounds. So compounds are formed when elements react. Compounds are substances formed from two or more elements, and the atoms of each are in fixed proportions throughout the entire of the compound. It's usually difficult to separate the original elements of a compound out again. A chemical reaction is needed to do this. So a compound which is formed from a metal and a non-metal consists of ions. The metal then loses electrons, the non-metal gains electrons. So what happens is there is a balance of electrons, but we'll look into that more in detail later. But an example of a compound, CO2, H2O, two common examples. Now we need to know and look at separation techniques. So here I always find it really simple to define each separation technique. So simple distillation is when you're separating a solvent. So a solvent is a liquid that dissolves within a solution from a substance. So what you do is you heat it at the boil. Both have two very different boiling points. You heat it at the boiling point of the solvent you want, and you can gain that in the end. Chromatography. This is the technique of separating different dyes in an ink with different solubilities. So you have your beaker, and you have your water in the beaker. You put your chromatography paper in there, you draw the pencil line just above the water, and then you put the spot of inks on there. If it's a different type of ink, it wouldn't move with water, you would use ethanol solution instead. And you can use something to stick this on top of, and then in the end, you get a nice chromatogram in which you can calculate the different lengths achieved. Then we have filtration. Now, filtration you may have used before in loads of different things. So it's when you have filter paper, and the filter paper collects this insoluble solid from the solution here, and it leaves the whatever else is in there. So if you wanted to get sand, for example, because sand is insoluble in water, you would use filtration, pour the sand and water solution into the conical flask, pour into it the filtrate, and then the water will be left. So that's quite the simple one. We've talked about that. Now we're going to talk about evaporation and crystallization. So this is used when you want to get a soluble solid from a solution. So what you do is you place the solution in a beaker, place the solution into a beaker, use a Bunsen burner to heat it, and then you're heating away the solution at the solution's boiling point, so that eventually you'll just end up with one of the elements and the intended element that has been that is soluble. So this works for salt, NaCl, and many other ones too. Okay, that's enough of the separation techniques. Now it's important to look at the history of the atom. So the history of the atom starts with in the early 1800s, the first thing that was discovered was, one second, sorry, getting lost here. So the first thing that was discovered was by John Dalton. I could not remember his name. So we've got John Dalton. And John Dalton said that everything is created by invisible little balls, solid spheres that make everything and define everything. Then we had J.J. Thomson. So John Dalton, J.J. Thomson. And J.J. Thomson discovered that the atoms actually weren't solid spheres. He measured charge and mass and showed that an atom must contain an even smaller negatively charged particle called electron. So he did not discover that the nucleus was positive. J.J. Thomson discovered the existence of electrons. So he found out that there's a smaller thing, because John Dalton said the smallest possible unit in the world is an atom, and J.J. Thomson said no, there's a smaller unit within this atom which he called an electron. So he then came up with the plum pudding model, that because he knew of the existence of electrons, but he also knew that the charge of an atom is positive. That an atom had to be positive is that there had to be a positive charge there as well. So he's described the atom as a model, saying that it's like a plum pudding cake. It's a sphere of positive charge with negative electrons embedded into it to create an overall neutral charge. Now, Ernest Rutherford conducted his alpha particle experiment, and he then found out, let's look at what he found out. So what Ernest Rutherford did was he fired positively charged alpha particles at a thin sheet of gold. Now what he did was he expected most to go through, because he realized that they are very small. And this prediction was correct. Most did go through, which suggests the very small nature. However, some were slightly deflected. And this meant that the center of an atom must be positive, because when the center got in contact, the center, which was positively charged, got into contact with the gold, it slightly deflected because like charges repel. Free science lessons video on this is really useful. I would suggest going and watching that. And then some were completely hit away and like rebounded. And the fact that some completely rebounded suggested that the center must be a lot of mass for it to completely rebound like that. And Rutherford came up with an idea to explain all of this evidence. He called it the nuclear model. So from this, he created the nuclear model. Now, the nuclear model suggested that there's a tiny positively charged nucleus at the center where most of the mass is concentrated, and then there's a cloud of negative electrons surrounding this nucleus. And they're just randomly put in. Now, we know this is not entirely correct because Niels Bohr then came up with the idea that there are a certain amount of electrons on different shells and energy levels. So we've come up with the idea of 2, 8, 8. And then I think it's 10. And then finally, James Chadwick came up with the idea of neutrons' existence. So just to summarize in the alpha particle experiment, when alpha particles came near the concentrated positive charge of the nucleus, they were deflected. If they were fired directly at the nucleus, they were deflected backwards. So this suggested that most of an atom is empty space as well. Okay, so that's the history of the atom revised. And now we just need to do a bit more on the periodic table. Metals, non-metals, group 1s, 0s, etc. So let me just take a note of this. Okay. So I didn't have time to write notes about this topic because I was trying to fix the stream. However, we can go over it now. So let's look at the periodic table. So the periodic table is arranged very specifically. In terms of the history of the periodic table, originally we see the law of octaves. And the law of octaves suggested that elements were arranged in groups of 8, those with similar properties. However, this was quickly proved wrong. And the reason why it was quickly proved wrong was the fact that when there were new elements that came, they did not fit the patterns. So therefore, his theory. Then we have Mendeleev's theory. Now, Mendeleev's theory was pretty much correct. So Mendeleev's theory was arranging the elements of the periodic table in order of atomic mass. Now, this order of atomic mass, however, did switch. So Mendeleev did switch this order of atomic mass when he realized that the properties should be changed. So when he realized that when arranging atomic mass, some signs of groups started to form as there were elements with similar properties, he then switched them to make the groups more defined. He also left gaps in the table for future elements to fit. And the reason why we now agree with Mendeleev's theory is the fact that each of the future elements and each of the predictions from Mendeleev as where we would put them has perfectly fit. However, there is now a reason why we don't use atomic mass and do not arrange the periodic table in terms of the atomic mass. Now remember, let's look at this. So the atomic mass is the number of protons plus neutrons. Now the issue here comes with neutrons. What used to happen is scientists used to identify loads of different isotopes, which means different numbers of neutrons, and therefore the positions in the periodic table were different. Therefore, instead of arranging it in terms of atomic mass, we now arrange it in terms of atomic number, which is the number of electrons. So that's the history of the periodic table. Now talking about the modern periodic table, there are roughly 100 elements which are all made from different things. So it's important to note that elements with similar properties form columns. So we have different groups. So groups form the columns, and each group has similar properties. And then the rows are what we call periods, hence the name. Now we definitely see a split. Over here is the staircase. So every element on the left side of the staircase is what we call a metal, and every element on the right side of the staircase is what we call a non-metal. Now, an easy way to remember this is that there are a lot more, just knowing the fact that there's a lot more metals than non-metals, therefore metals will always have the advantage over the periodic table. Now, there's a lot more to these groups and rows than you may think. So the groups tell you the number of electrons on the outer shell. So in group 1, there is 1 electron on the outer shell. Now the rows tell you how many rows will form, how many electron shells there will be. The exception to this, however, is group 0, for helium has 2 electrons in its outer shell. This is useful as the way the atom reacts depends upon the number of electrons in the outer shell. So the fact that it's arranged in terms of the number of electrons then tells you the reactivity and the properties. So most elements are metals, as we said. Metals form positive ions. What does this mean when we say metals form positive ions? So metals therefore have too many electrons. The whole goal of these elements are to form a full outer shell. And metals always seem to have 1 or 2 or 3 or 4 extra on the outer shell, such as, let's look at lithium. Lithium has 1 on the outer shell. So therefore, what happens is these metals get rid of these electrons. Now we know that electrons are negatively charged. So if we're getting rid of a negative charge, then you're becoming a positive charge. In this case, positive 1, because you've got rid of 1 electron. If you got rid of 3 electrons, you will form a positive 3 electron charge. And when they become charged, we then call them ions. So looking at the different properties are really important as well. Metals have specific properties. So metals' properties tend to be strong and hard. This is except for alkali metals, which are quite soft. And they're strong and hard, and they can be bent and hammered. They have high melting and boiling points. And they can also conduct heat and electricity quite well. We also have, so when we discuss metals, we mainly are discussing either the alkali metals or this big group here, which we call the transition elements. And a vast majority of these transition elements are in fact metals, things such as iron or titanium. Now, transition metals may have more than one ion. The reason why they're in this weird group is because the number of ions, so the charges they form, are different. So they're almost these wishy-washy elements, and that's why they're put in this block. Some of them are typical metals and have the properties of a metal. Some of them are like really soft. Mercury is really cool, and they are coloured compounds as well, so they're quite colourful. They're also used as catalysts, which speed up reactions. For example, nickel is used and iron is used as catalysts. Finally, we're going to just look at the periodic table elements now. So, let's look at the key groups that you need to know. The first key group is group 1, then you need to know about group 7, and finally group 8 or group 0. So group 1 are called the alkali metals. The reason why they're called alkali metals is because when they react within a solution of water, they form hydroxide ions. Now, the alkali metals are lithium, sodium, potassium, rubidium, cesium. So you don't need to know much below that. So a key fact here is that as you go down the group, the reactivity increases. Now, why is this? So all of these, being in group 1, have one electron on the outer shell, and their aim is to get rid of this electron to form the positive ions. Lithium has one shell between the nucleus and the outer electron. However, potassium has a lot more shells, and as you go down the group, the number of shells increase just because the elements increase. Therefore, the distance between the outer electron and the nucleus is much more in potassium than it is in lithium, who doesn't want the electron to go away. So in lithium, clearly, because the mother is physically closer to the electron, it's harder for it to go. However, in potassium, because it's separated by loads of layers, the mother is more distant from the electron, and therefore the electron is more willing to move, and therefore as you go down the group, there is increasing reactivity. Now, you may be asked this question a lot: why does it increase in reactivity here? And this is exactly what you need to say. Now, we're going a bit over in terms of time, but thank you to everyone who's watching right now. Just want to say, feel free to put them in the chat. So just to go back to group 1. Alkali metals tend to be light. They float on water. They basically subvert your typical connotations of what you would associate with a metal because they float on water. Well, the first 3 at least, they're quite low density. They have low melting points. They're quite soft, and they react quite well with water, chlorine, and oxygen. Now let's look at group 7. So group 7 are what we call the halogens. So the halogens are all non-metal elements. So they all end in "-ine", so fluorine, chlorine, bromine, astatine, or iodine and then astatine. So you can think of group 7 as the opposite to group 1. Group 1 are made of metals that all have one outer electron. They want to get rid of this outer electron to form a positive charge. Group 7 elements all are nonmetals. Nonmetals that have 7 outer electrons. Now, in this case, they want to form a negative charge and gain 1 electron to form a full outer shell. Now, as we know, as you go down, the number of shells increase as you go from the nucleus. So there's a lot more going down the group. And the nucleus is in the center. And in order to get a full outer shell, the nucleus needs to attract an electron. However, as you go down the group, the distance between the nucleus and the electron would only increase. And as the distance increases, this then means that as the distance increases, this means that the reactivity decreases. Because it's harder when there's a lot more shells for the nucleus to attract that extra electron. Now let's look at some properties of group 7 elements. So group 7 elements have higher melting points and higher boiling points as you go down the group. Why is this? So as you go down the group, it's harder to react. Therefore, and there's more shells, therefore there's a higher melting boiling point. There's higher atomic masses, so they're actually heavier and quite dense. And it also gets darker, so you need to know the colours. So in fluorine, it's quite green and yellow, and then it gets quite dark and brown and quick really easily. Now halogens form molecular compounds, so they share electrons via covalent bonding. So if we look at fluorine, for example, we always see these as with a small 2, and the reason why we see them with a small 2 is because they form a molecule, a covalent molecule. So they share electrons with each other. So the fluorine, for example, shares electrons with each other because they are quite unreactive, so they want to create their own reaction. And that's why we always see them as molecules with 2. Now, they also form ionic bonds. So they form covalent bonds with themselves. Covalent is between nonmetals. They form ionic bonds with metals in group 1. And they react to form halides. So the alkali metal sodium and the halogen chlorine can react to form sodium chloride, salt. Oh, thank you, Livia, for your comments. It means a lot. I'm really grateful for you. Okay. I'm glad this is helpful. If you want me to go over anything, I can do. Just feel free to tell me. So, we've gone over group 7 elements. And finally, we're going to go over group 0 elements. So let's look at group 0 here. So you may be wondering, why is it called group 0? So group 0 elements are what we call the noble gases. They are unreactive. They do not react with anything. Why do they not react with anything? They have a full outer shell. And because they have a full outer shell, this means that they do not have to become charged to gain a full outer shell because they already have it. So they do not have to form ions. Now, they still have properties such as the boiling point increases as you move down the group, and there's more electrons as you move down the group. So we see helium, neon, argon, krypton. So as you go down the group, the melting point and boiling point increase because there is more electrons, and as there are more electrons, this means there's a greater intermolecular force between them, which needs to be overcome. Therefore, more energy needed. Now, In terms of properties, they exist as monatomic gases. So this is the opposite of what we saw in group 7 when they share electrons. So when we see F2, for example, we will never see helium 2 because they are monatomic. They exist by themselves. And they also are colorless gases and non-flammable. They will not set on fire. Okay, so that is chemistry. If you've missed anything, feel free to note that down. Now we're going to look back onto our next topic, which is bonding, structure, and properties of matter. So I want you to write as much as you can remember within this last 8 minutes. I'm sorry this is running over massively, but within 8 minutes up till 7 o'clock, please write as much as you can about ionic covalent bonding, polymers, metallic bonding, states of matter, and nanoparticles. You get a lot of questions about these, so we're going to go through that in a second. See you soon. Hello, I think hopefully you can still hear me. I'm unsure if you are able to, but we're out of time for this topic, unfortunately, but I'm going to hopefully try and speed it up and give you a quick summary of the topic. Hopefully you can still hear me. Let's now go through this topic. So, bonding structure and properties of matter. So I'm just going to call this topic bonding. Now we're first going to look at ionic bonding. So ionic bonding is when elements react in order to achieve a full outer shell, and this is when a metal reacts with a non-metal. The metal then forms a positive ion, and the non-metal then forms a negative ion, and it's a win-win situation for both parties. Now, this is quite simple, and in terms of properties, of ionic compounds, because these are quite important, what happens is it forms a giant ionic lattice. So this giant, thank you, Olivia, for the confirmation. For this giant ionic positive ion is surrounded by a negative ion. So imagine sort of formation. So we have the bonds in between. So you can see how each positive ion is connected to each negative ion. And it then forms a three-dimensional structure. Now we can link this to our knowledge of salt. You may be like, how can salt be linking to this? Well, we know that salt is a crystal structure, and salt is formed from sodium and chlorine. Sodium being the metal and chlorine being the non-metal. Therefore, sodium chloride is an example of a giant ionic lattice with the three. So hopefully that may help you in terms of learning that. Now, we need to know about these bonds here. So these bonds are very strong. These ionic bonds are very strong. Why are they very strong? Because of the fact that there's a very good force of attraction between the negative and positive charges and ions. So there's a very strong electrostatic attraction. And this means that certain properties with giant ionic lattices, such as the fact that there's a very high melting point and boiling point. Now, this very high melting point and boiling point is because of the fact that there are very strong forces of attraction and they require a lot of energy in order to overcome them. Now, the second property is the fact that it cannot conduct electricity when solid. Which makes sense in our minds, right? Because you don't assume that salt can just conduct electricity. So salt cannot conduct electricity as a solid, but if it's molten or if it's dissolved in water, then it can conduct electricity. And the reason why it can conduct electricity is because there are delocalized electrons. Delocalized electrons are when electrons are free to move around and it can therefore carry a charge and therefore conduct electricity. So they're the properties of a giant ionic lattice. Now we need to know about covalent bonding. So we saw that ionic bonding is between a non-metal and a metal. Covalent bonding between non-metals only. This is exclusive, and it's normally when non-metals share electrons between themselves. So that's what it is. Covalent bonding is when non-metals share electrons to form a full outer shell. So we have simple covalent bonds, which is the first variation of covalent bonds. So simple ones are things like hydrogen and water. So you have two hydrogen molecules connected to one oxygen molecule. Now these are all non-metals and they are all sharing electrons. And this is an example of a simple covalent bond. Now sometimes we can get double covalent bonds. Now, what are double covalent bonds and where do we see them? So double covalent bonds is, for example, in oxygen. So when oxygen, because oxygen is in group 8, or it needs 2 electrons to form a full outer shell. And therefore, it needs to have 2 covalent bonds to therefore share these 2 outer electrons. Now, double covalent bonds don't tend to be asked about, so it doesn't matter as much if you don't understand it. Just be aware of the fact that double covalent bonds exist, and it's just when non-metals share more than one pair of covalent bonds. Now, these are all relatively simple covalent bonds, but we also have structures called giant covalent bonds. But before we move on to giant covalent bonds, I just want to say that with simple covalent bonds, we also have specific properties that you may need to know. So properties of small covalent bonds goes as follows. So properties of small covalent bonds. Firstly, there's a low melting point and there's a low boiling point. This means that things like water and oxygen tend to be liquid or gas at room temperature, which we know. We know that oxygen tends to be gas. That's how we breathe it in. So the reason why we have this is because if we look at the structure of water, for example, what we see here is that these bonds, these intermolecular bonds between these intermolecular covalent bonds connect oxygen to hydrogen are very, very strong. And they're very strong because they're sharing the electrons. However, also, apologies, let me just write that again. So these bonds are what we call the covalent bonds, as we discussed. So they're the bonds between hydrogen and oxygen, and these are really strong, as we said, because they are sharing electrons. However, these bonds are intermolecular bonds. And these intermolecular bonds are very weak, which means that because they're very weak, when there's heat applied, it very easily breaks apart into a structure that's normally a liquid or a gas. But it doesn't mean that the molecules themselves are weak. It's more the bonding between the molecules that are weak. I hope that makes sense. Okay. And the next property of small covalent bonds is as the size increases, the intermolecular forces increase. So what this is saying is as the number of covalent bonds and sizes, a number of covalent bonds between these increase, then the intermolecular bonds also increase. Now, the final property of small covalent bonds is that they do not conduct electricity. Now, the reason why is because they do not have an overall electric charge. There's no overall electric charge because of the fact that the metal, which is positive, cancels out the negative non-metal ion, therefore forming an overall charge of neutral. Therefore, there's no electric charge. Now, finally, moving on to the giant covalent bonds. So, giant covalent bonds are always solid at room temperature. As they have millions of strong covalent bonds. So as we discussed here, we said that with the size increasing, the intermolecular forces increase. So how this affects this one is with giant covalent bonds, giant is not in reference to the size of the molecules, but more the amount of molecules. So because there are millions and millions of covalent bonds, this means that it's always solid at room temperature and things like that. So it's always solid at room temperature. And there are 3 key giant covalent structures that you need to know. So the first one is diamond. Then it's silicon dioxide, which is also known as sand. And finally, graphite. So, in terms of diamond, diamond is formed from the element carbon, which, as we know, firstly meets the criteria of being a non-metal. So diamond is formed from carbon, and each carbon atom forms 4 covalent bonds with other carbon atoms. So key number here, in diamond we see 4 covalent bonds between the carbon atoms. Because there are 4 covalent bonds and carbon has 4 electrons that they need to share, this means that it fully reacts, and therefore there are no delocalized electrons. There are no spare electrons because they're all being used for bonds, and therefore no electricity can be conducted. And this is the strongest structure on Earth. Diamond is the strongest structure on Earth because of the fact that there are 4 covalent bonds and there are 4 extra electrons. So everything is reacted perfectly, so that it's so perfect that it doesn't want to separate. So that's why carbon is so perfectly reacted. It doesn't. No electricity can be conducted, and the melting and boiling point is super, super high. Okay. Now onto silicon dioxide, which is sand. So silicon dioxide is between silicon and oxygen. Now silicon and dioxide basically tells you that there's 2 oxygens. And what happens is, all you need to know is that there's a high melting point, high boiling point. Because of reasons, you can quite assume that because it's a high melting point and boiling point, it must mean that everything is reacted perfectly and there's covalent bonds are very strong, and it does not conduct electricity. if sand did conduct electricity, we probably wouldn't go on them on beaches. So just use that to help you remember. And finally, graphite. Now, graphite is, like a diamond, also formed by carbon. However, there's a difference. Each carbon atom only forms 3 connections. 3 is a number here. I don't know what that connection is. So each carbon atom only forms 3 covalent bonds. Now, as we said, diamond is perfect because 4 covalent bonds are needed for carbon to react perfectly. However, we only have 3 here. This then means that we have 1 electron spare, and that's not being shared. And what do we call a spare electron? We call it delocalized. And when there are delocalized electrons, what does that mean? That means that there are free electrons available to carry charge. And therefore, graphite is able to conduct electricity. That's why graphite is very useful. So graphite, because it's in its 3 carbon bonds, they then form hex layers. So you can imagine 6 carbons, and each of them form 3 bonds. So they form hexagonal layers. I don't know why I was attempting to draw that. Hexagonal layers. Hexagons are very slippery. And because they're slippery, this means that graphite can therefore slide. And that's why we have graphite within pencils. What we call the lead of the pencil is actually formed from graphite, and because it is able to slide, that's how we're able to write on paper, because the graphite is sliding. Now, the reason why it's able to slide is because there are no covalent bonds between the layers. Whereas diamond obviously has covalent bond between the layers. We do not see this in graphite. Now, those are the 3 giant covalent structures. Now we need to look at graphenes and fullerenes. And then we're going to finally finish on metals and alloys. This is sadly overrun into the physics sessions. Therefore, I'm very apologetic. But hopefully we'll have time to look over that too. So let's look at graphenes and fullerenes. So what is graphene? Graphene is a single layer of graphite. So remember, we said graphite are these hexagonal layers of carbon that form 3 covalent bonds and therefore can conduct electricity. So graphene is a single layer of graphite. It's an excellent conductor of electricity, as we said, and it's extremely strong. So what are fullerenes? Fullerenes are molecules of carbon atoms with hollow shapes. So molecules of carbon with hollow shapes. Now, usually fullerenes have hexagonal rings, but they can also have rings of 5 carbon atoms or 7 carbon atoms. So we tend to go around those 3 numbers. Now, there's a special fullerene that you need to know, which is called the Buckminster fullerene. It's one word, I assure you, and it's probably one word you will not forget because it's very long. So the Buckminster fullerene contains 60 carbon atoms. And these carbon atoms therefore arrange to form a hollow sphere. Now, we can use the Buckminster fullerene in everyday life. So they tend to be used in terms of the pharmacy. They're used for the pharmaceutical delivery of drugs. So they normally house drugs and take it into your body and then release it when ready. It can also be used as catalysts. It can also be used in things like sunscreen and things like that. Now, finally, we're going to look at metals and metallic bonding. Okay. So, metallic bonding, as you can guess by the name, is when there are bonds between metals. Now, metals consist of a giant structure of atoms arranged in regular layers. So, metals are giant structures, yet again. Metals are giant structures of atoms arranged in regular layers. And each of the regular layers contains electrons on the outer shell, which are delocalized. Now, the metallic bonding is the bonding between the strong electrostatic attraction let me zoom in for you. So metallic bonding is a strong electrostatic attraction between the sea of delocalized electrons and the positive metal ions, because we know that metals form positive ions and then also have delocalized electrons, and this then creates a metallic bond. There's high melting points and high boiling points. They're excellent conductors of heat and electricity. And metals can be bent and shaped into things like alloys. It can be distorted, and in order to make metals harder, you can make them into alloys by distorting the layers. And when you distort the layers, this makes it difficult to slide over each other. And therefore, this then means that it's a stronger metal. So if you were ever to be asked a question, which I've seen before, is why are alloys stronger than metals? You'll say alloys are distorted layers of metals, and because it's distorted, they are then difficult for the electrons and elements to slide over each other, and therefore it's a stronger metal. Now, finally, the final topic is nanoparticles. Nanoparticles are these really small particles that have a very small diameter and contain thousands of particles. So these particles have a very high surface area to volume ratio. So you've hopefully done this in biology, in which the surface area to volume ratio is really useful, because the higher the surface area to volume ratio, the more can be absorbed for the same volume. So the more can be absorbed for the same volume. And that's why humans have to have all of these adaptations, such as alveoli in the blood, in the lungs, because of the fact that we have a low surface area to volume ratio, so we need to create added surface area. So because of the high surface area to volume ratio, nanoparticles are really useful. They can be used for things such as medicine, cosmetics, catalysts, and there are potential risks such as nanoparticles being absorbed unknown. But that's the final topic for structure and bonding. Well done for getting this far. Okay, we're now going to go through physics. Luckily today is the first day as a trial, so we can do physics, all of the first topics, which tend to be quite a lot easier, before tomorrow, and the next day we'll be going on to the harder topics. I'm just trying to get to the physics. Okay. So now moving. Okay. It was supposed to start at 7, very much apologetic for that. We kind of slightly run over, but good, it's good. Revision is good. Okay. So the first topic I would like you to do again is energy. So I want you to blurt as much as you can on these 9 requirements here. I'm going to give you probably 5 minutes because this topic is really small anyway and there's not much you need to say. So I'm going to give you till 7:32 to write as much as you can about energy. Okay, so let's begin by revising this short topic. So, in terms of this specific topic, in kinetic energy, gravitational potential energy, elastic energy, there's a lot of equations. However, I do know that for your year, if you're in year 11, then that means that you're going to be given the equations. So, if you're given the equations, I think it's still useful to know about the equations so that you can easily look back on them. And however, I'm not going to pay too much attention to it. So let's begin. Look at energy. So the first thing we're looking at is energy stores and systems. So energy is something that I'm sure you've heard, the law of conservation of energy. That energy is neither lost nor made. So energy is not created or destroyed. It can only be transferred. So energy is transferred. And when energy is transferred to an object, this energy is then stored in the object's energy stores. So there are 8 different types of energy stores. Let's see if we can name all of them. So let's say thermal energy, elastic potential, gravitational potential, kinetic, chemical, magnetic, nuclear, and electrostatic. So these are the different types of energy stores that you need to know. Now, energy can be transferred through 2 different ways. Energy can be transferred mechanically, or energy can be transferred electrically. So, what does that mean? Mechanically means by doing work. Now, work is anything that requires like energy physically. So, if you move your arm up, that's work done. And the other ways energy can be transferred is through heat, which is heat or sound. Heat or sound. And both of these are referred to as radiation. So, energy can be transferred through either mechanically or through radiation. Now, radiation can be heat, light, sound, etc. So, in terms of physics, I know what would often confuse a lot of people is the idea of there being a system. So, what is a system? A system is when you refer to in physics, things that come into and out of so system is sort of the objects that you're interested in, whatever objects you're interested in. So say if you wanted to investigate a ball in the air, then your system will be the ball and the air. If you did not want to look at the impact of the air, then you'll have to do it in a vacuum where there's just the ball. So a system is a fancy word for the objects that you're interested in. Now, a closed system is when neither matter nor energy can enter the system or leave the system. This is what we call a closed system. For example, if you look at space, space has no air. Therefore, no energy or matter can be transferred in because energy and matter can be transferred through air. But if there's no air, then that's not possible. So that's an example of mechanical. So when we're looking at mechanical energy transfers, they tend to talk about systems. When they're talking about radiation, then they tend to be talking about when you're boiling a kettle and how water moves up and how temperature increases and things like that. So work done is just another way of saying energy transferred. They're the same thing. So when you say I've transferred energy, that's the same thing as saying I've done work. But work done is not only when you're physically doing something, work done can also happen if there's a current flowing. So work done has two things. Firstly, when energy is transferred. And secondly, when a current flows. Because when a current flows, there's going to be resistance. Resistance is how difficult it is for current to flow. And if there's difficulty, that means energy is being used to try and go against that resistance, and therefore work is being done. Now we need to look at kinetic energy and potential energy stores. So kinetic energy is the energy stored in a moving object. So when an object speeds up, energy is transferred to the kinetic energy store, and energy is transferred away from the kinetic energy store when an object slows down. The energy in the kinetic energy store depends on two things: mass, which is how much something is made of, and the speed. It's kind of logical if you think about it. If you're thinking about how much energy you have in your movement, you have to think about your mass and your speed. And that's where we get the equation that kinetic energy is equal to half times mass times velocity squared. But of course, you're going to be given that in the exam, so don't spend too much time revising that. So we have kinetic energy. Now we have potential energy. So potential energy is the energy in between the particles, and we have different examples of potential energy. So there's elastic potential energy, gravitational potential energy, which is what we're going to look at now. So gravitational potential energy is things like when you are lifting an object. So when you lift an object, you're then lifting it within a gravitational field, and therefore you're doing work, and therefore energy is being transferred, and therefore there is a gravitational potential energy system. So the higher that this object is lifted, then the more energy is transferred to this store. Therefore, if we're looking at what affects the gravitational potential energy, height affects it, because as you go, the further you increase in height, the more gravitational potential energy there is. Mass is also important. And finally, the gravitational field strength of you are. So on Earth, it is 9.8, but on different planets, it's different things. That's why we don't just put times 9.8. Now, falling objects also release gravitational potential energy. So you don't only have gravitational potential energy when you are moving something up, you also have it when something is falling down. So say if we had a bungee jumper, because a bungee jumper is normally used in exam questions because it combines this idea of both kinetic and gravitational potential. So when we have a bungee jumper, as they fall down, and then they fall back up. There's also elastic potential energy in this as well. Okay, so as they fall, energy from its gravitational potential energy is then decreased. So as it falls down, the gravitational potential energy decreases. But what does increase is the kinetic energy because there's movement occurring. So the kinetic energy increases. Now, the person continues to fall down, and as you continue to fall down, the gravitational potential energy continues to decrease, but the kinetic energy continues to increase. However, when you reach the bottom here, the rope begins to tighten, the bungee jumping rope. So as the rope begins to tighten, we see something called elastic potential energy, because then from this point on, the rope is now stretching beyond its limit. So therefore, energy is being transferred from kinetic energy to, so the kinetic energy decreases and the elastic potential energy increases. Now this continues until the person gets right to the limit of elasticity, when the rope tightens very much. And this means that all of the energy at this point, at this lowest point, is all elastic potential energy. But now the person goes back up and we see the opposite happen. So as the person goes back up, the elastic potential energy decreases, the kinetic energy then increases, and then the kinetic energy, because you're moving upwards, the kinetic energy then decreases, and the gravitational potential energy then increases. However, you're often asked in an exam, why is it that as the person jumps back up, the person never jumps to the full length as where they jumped from? So if a person jumped from here, why do they never end up bouncing back, rebounding to that same height? Why do they end up only rebounding here? And you can say energy is lost in friction with the air, and friction with the air is called air resistance. Now we've discussed the gravitational potential energy and kinetic energy, but just so you know the elastic potential energy equation, elastic potential energy is half times K times extension squared. So what is K? K is what we call spring constant. So it's just a value that determines for a spring. It's a value that determines for the spring the limit of its proportionality. And the extension is how long it's been extended. And there's a required practical regarding elastic potential energy, which we will look at soon. Okay, so that's energy stores. We're now going to look at specific heat capacity. So specific heat capacity has quite a long-winded definition for you to know. So specific heat capacity is the amount of energy required to raise the temperature of a substance by 1 degree Celsius exactly for 1 kilogram of that substance without changing the state of the material. So the energy required to increase the temperature of 1 kilogram of substance by 1 degree Celsius without changing the temperature. Because it's energy required, specific heat capacity is measured in joules. So if we're looking at the value for water, the specific heat capacity for water is 4,200. Now, you don't need to know this off by heart because you'll be given it in the question. But what we're saying here when we mean the specific heat capacity of water is 4,200 is that 4,200 joules of energy is needed to increase 1 kilogram of water by 1 degree Celsius without changing the temperature. Now, specific heat capacity of a material like mercury, for example, is 139 joules. So this means that only 139 joules of energy is required to increase 1 kilogram of mercury by 1 degree Celsius. So if we were to compare these two materials, what does this suggest? This suggests that mercury is much to heat. And the reason why it's easier to heat is because it requires less energy. But this also means that because water requires more energy, it sustains this energy for longer. It sustains this energy for longer. Now, what that means is it keeps hold of the heat for a longer period of time. Because mercury is so easy to heat up, it also means it's easy to cool down. Whereas water takes a long time to cool down. Therefore, that's why we see water being used in heating and things like that. So water is used in heating because it keeps hold of the heat for so long. It's used in things like radiators. Now, you can actually investigate the specific heat capacity because there's an equation. And the equation luckily involves all of the things we talk about within the definition. So increase in temperature, 1 degree Celsius, 1 kilogram. So in terms of looking at the equation, specific heat capacity is often written as specific heat capacity is one of the variables. So the change in energy is equal to mass times specific heat capacity times the change in temperature. By the way, if you didn't know, this triangle means change. So change in whatever value. So therefore, if we were to draw it in a triangle, the change in energy is the mass times the specific heat capacity times the change in temperature. So if you wanted to find the specific heat capacity, you would do the change in energy over the mass times by the change in temperature. But again, you're given this equation, so don't worry. And as we said, you can investigate the specific heat capacity of a substance by using certain equipment and I believe it's a required practical. Yes, it is required practical. So let's look at this required practical. What you're doing in this required practical is you're investigating the specific heat capacity of a block material. Now, we are already aware that the specific heat capacity is the change in energy over mass times the change in temperature. So, in order to find the specific heat capacity of a material, we then need to find the change in energy, the change in temperature, and the mass. Now, firstly, we can easily find the mass by weighing the material. Let's say we're finding copper. So weigh the mass of copper. And then what you need to do is for this required practical, you're going to be given a specific block of copper in which there are 2 holes in this copper. Oh, experiencing some internet issues, sorry. Okay, so in this copper block, there are 2 holes. Now, in one of the holes, you're going to put the heater, and in the other hole, you're going to put the thermometer. We'll explain why in a second. So the heater, in order to work, needs to be connected to a circuit. So there's the power supply. And we also have an ammeter, which controls the amount of heat. So what we'll be doing is first weighing the copper. Now, normally your teacher might tell you just how much it is. It might just be 1 kilogram specialized, or you may have to weigh it. And then what you want to do is you're going to wrap this in an insulating layer to reduce the energy transferred, because you want the amount of energy being transferred to be calculated to be as correct and accurate as possible. So in order for the change in energy to be as accurate as possible, you then want to make sure you've got an insulating layer. So imagine this red thing is a bit of newspaper around it. Insulation. Okay. Now what you're going to do is you are going to insert the thermometer and start a stopwatch. You're going to measure the initial temperature of the block. So you need to measure the initial temperature, because remember, we're looking at the change in temperature. And then what you're going to do is you're going to switch on the power supply. And when you turn on the power, the current in the circuit works on the heat transferring energy electrically, changing the thermal energy store. Now, the reason why we have an ammeter is so we can measure the change in energy. Energy and measure the change in energy. So the initial energy will obviously be 0 because there's no energy in the copper block to begin with. And the change in energy is how much energy you have supplied to the material. So it will be X value. And now we have the change in energy, the change in temperature. The change in energy, the mass. We need to work out the change in the temperature. So, and you then record the final temperature and deduct this away from the initial temperature. Now, when do you stop? When do you take the temperature? So you take readings of the temperature and the current every minute for 10 minutes. So it's going to be, you're going to have a table. So minute 1, for example, minute 2. In each one, you have the current reading, you have the temperature change, or just the temperature, and then you can work out the change later. And that's it. And the mass is going to be constant throughout the whole thing. And then you can eventually work out the specific heat capacity using some calculations. So the y-axis would be the temperature, and the x-axis would be the energy transferred. And the graph should look something like this. So as the energy transferred increases, the temperature increases. Okay, so that's enough of that required practical. Let's look at what we have to revise next. So we've revised specific heat capacity, conservation of energy, conduction and convection, and renewable energy. Okay. We have 4 minutes left, so I'm going to try and make this as rapid as I can. So conduction and convection are 2 ways in which thermal energy is transferred. Conduction, convection. Conduction is where vibrating particles transfer energy to neighboring particles. Vibration, this allows particles to transfer energy. Now, in order for particles to vibrate, they must be solid. So conduction mostly happens in solids. So the vibration of particles, which therefore transfers energy. Now, this energy is shared across the kinetic energy stores of the particles of the object. So although we're transferring thermal energy, vibration therefore means kinetic energy of motion. The particles in the part of the object being heated vibrate more and collide more with each other. These collisions cause energy to be transferred between the particles' kinetic energy stores. So conduction is transferring thermal energy through the kinetic energy stores of vibrating it. Now, this process continues throughout the object until the energy is transferred to the other side of the object. So, say if we have whatever's heating the hot end and the cold end, the conduction continues all the way until the energy has reached the end of the cold side. Now, thermal conductivity is a measure of how quickly energy is transferred through a material. So, thermal conductivity, how quickly energy is. Sorry for this. A bit of issues here. Last minute issues as everyone starts using the internet. So, how quickly energy is transferred. Now, convection is when energetic particles move away from the hotter to the cooler regions. So, convection is energetic particles moving away. Now, if you study geography, you will know about convection currents, and that's how that happens within the mantle and heats the world up. And it can happen in gases and liquids. So it can happen in fluids. Energy is transferred by heating to the thermal stores of the liquid or gas. Again, this energy is shared across the kinetic energy stores of the gas or liquid's particles. So both of these have this similar idea that what happens is the thermal energy, is transferred into kinetic energy, which either causes the particles to vibrate and energy to be transferred like through conduction, or causes the particles to move away through convection. Now, because liquids and gases can flow, the warmer and less dense regions will then rise above the cooler regions. So the hotter particles, because they have more energy, they're then going to rise up and spread out, whereas the cooler particles become more compact and go down towards each other. Now, radiators create convection currents. So convection currents, as we talked about, is a key part of geography. Heating a room with a radiator relies on convection currents. What happens is energy is transferred from the radiator to the nearby air particles by conduction. So we get heat is transferred from the radiator to the air. Then what happens is the air by the radiator then becomes warmer and moves up. And therefore spreads into the room. This warm air then rises and is replaced by cooler air. The cooler air then comes back down as heated again. So when it's heated, it rises, and when it's cooled, it comes back down. And this circuit creates heated air, and the cycle repeats, causing a flow of air to circulate around the room called a convection current. Now, all you need to know about the other topic is that efficiency is equal to total power output over total power input. So, but you'll also be given that equation. And the rest, renewable energy sources, they're quite self-explanatory. Renewable means that they are sustainable. And yes, so that is the end of this morning live stream. Well done, Livia, especially for you for staying awake and alive and watching these live streams. And well done to anyone in the future who's gone through these. And well done, you've done your science revision for today. We'll be continuing at 9 a.m. doing English language, English literature, and maths. So see you then if you're going to be there. And bye for now.